Thank you very much, Joe. I should say it's really an honor to be the inaugural lecturer in this series. And, I just met Coleman Szostak for the first time, and I'd just like to thank him personally for endowing this series and for all that you've done for the university. I'm very new here, as you probably know, but I'm extremely happy to have joined the faculty here at the University of Chicago. It's a great place, and I love it here. So let's see, with that, let me dive into things here. As Joe said, we got interested in how life got started quite a while ago. We've made a few small advances, but it's a lot of big questions, so we're not done by any means. The reason that I got into this actually was we had been using laboratory-based evolution to make new and occasionally useful molecules. But that made me start to wonder, like, how did Darwinian evolution actually get going all by itself, on the early Earth, right? I mean, I'm sure in the end, we're going to say, it's got to have been simple, and why didn't we think of that earlier? But for now, we're struggling trying to figure out various parts of what we think happened. Let's see, what you see here is our somewhat out of date, but our imagination of what a very primitive early cell might have looked like, basically an extremely simple membrane. So yeah, we've spent quite a few years looking at the biophysics of simple membranes, and inside small bits of some genetic material, right? So for evolution and for life itself to get started, you have to have the ability to encode information, to propagate that, to have the variation that is what Darwinian evolution acts on. And so the question is, what were these molecules that were the basis of heredity in the very first cells? And so as you can see from the title of my talk, we think it's RNA or something, more or less like RNA. So why do we think that? And this is really the smoking gun. This is a picture of a crystal structure of the large subunit of the ribosome, which is the cellular machine that makes all of our proteins. This particular image is from Tom Stice's lab. And what you see here is, like, all the gray squiggles are RNA, and these gold squiggles are proteins. And this green thing in the middle is a transition state analog that shows you where peptide bonds are put together in the middle of this giant machine. And it's all RNA. So what you see from this image is that RNA makes proteins. All of our proteins, all of the proteins made in all of biology are made by RNA molecules. The ribosome is a ribozyme that synthesizes proteins. And so the only logical inference from that is that, okay, RNA must have been there first. And at some point during the evolutionary process of coded translation evolved. And that's kind of part of how we got to be what we are today. In addition to this, the whole process of coded peptide synthesis is mediated by lots of other RNAs, right? There's at least 50 or so transfer RNAs that bring the amino acids into this machine. So there's a lot of RNAs there. And then the information is in messenger RNA, okay? Transcribed from DNA, which is an archival storage system, but so you have another role for RNA. And in addition to that, what we've seen over the last 10 or 20 years of biological studies is just exploding list of things that RNA does in modern cells. So we have in bacteria, we have riboswitches, which play regulatory roles. In eukaryotes, we have microRNAs, we have long non-coding RNAs. It's like RNA is everywhere doing everything except for a few little things that proteins do. And I know some of you are interested in proteins, but that's okay. It all started with RNA. Okay, so that's fine, but you know, RNA is pretty complicated. Well, before we get into the complications, let me just go back to a simple model. So again, this is what we think of as a simple, very simple primordial cell or protocell. Okay, simple membrane, might be made of fatty acids or slightly more complex lipids, and this replicated this genetic material. And the things that have the puzzles that have absorbed us for the last 20 years are like how do these don't want to switch things off. How does the membrane grow, and how do you get division without any evolved biological machinery? And that turned out to be actually pretty easy. There are lots of ways that can happen spontaneously. The harder problem is how do you take some primitive genetic polymer and actually copy those sequences and then copy the copies and copy those copies so that you have a chain of heredity of information. And that turned out to be a lot harder of a problem, and we're still working on lots of parts of that. But it turned out that by working on that problem of how to copy and replicate RNA without enzymes, right, before there was any machinery, that's what actually ended up giving us, kind of as a byproduct, some insight into why all of this started with RNA and not some alternative genetic material, of which it turns out there are many possibilities. Okay, so how do we think about copying RNA without enzymes? So I want to show you this movie. It's a little bit out of date, but it'll give you the idea. So the idea is we have some short strand that's floating around in a very chemically rich environment, and you have highly reactive building blocks that are floating around. They find their partners pairing by Watson-Crick base pairing and gradually click into place, sort of like, you know, click chemistry that just won the Nobel Prize. The reason my voice is a little rough is because we were celebrating various things last night, including Carolyn Bertosi's prize. But yeah, that's how we were thinking about it. Let's see. Okay, so then you have to say, well, what kinds of building blocks actually could click together to make a complementary product strand, right? So first step in having heritable information is to make a copy of a strand that might encode some useful information. And so this question goes back at least 60 or 70 years. And the late Leslie Orgel is really the person who started serious research in this area. And he made many contributions, but to my mind, the most important thing that Leslie realized was that the molecules that we see in biology as substrates for copying RNA and DNA, the nucleoside triphosphates, are fantastic if you have enzymes to catalyze the reaction. Right? They're not very reactive. And so by themselves, they don't really do anything except sit there and slowly degrade, which is not very useful. Leslie realized that what you need to get life started were more reactive building blocks. Things like this, where the phosphate here is activated by this, for example, imidazole derivative that you see here. So these things are chemically much more reactive. And when they bind to a template strand, they will start to, like, very slowly and not very accurately assemble into a copy. Okay. They have other advantages. They're less polar, so they can get across membranes spontaneously. So we think that, you know, something like this is more reasonable for getting all of these processes started. Now, this particular leaving group, methyl imidazole, was something that was found in Leslie's lab in early 80s. And, you know, it kind of works. It's not great. There's no particular reason to think that it would be the actual thing in prebiotic chemistry, right? You can draw pathways to make this from simple chemicals, but there's nothing really special or unique about it. So it was always viewed as kind of a lab model, not necessarily anything actually involved in early copying chemistry. So about 5 or 6 years ago, we got interested in this, and Lee, who was a postdoc in the lab at the time, did a small screen of different substituted imidazoles, trying to see if he could see some trends and work out something that was better. And it turned out, after looking through a small collection, that this 2-amino imidazole is like much better. It's way better. And I'll show you an example of that. But this also gives you a kind of taste of how the field progresses. It only took 30 years to realize that if we change a methyl group to an amino group, things are better. So the pace is not always what you'd like it to be. Anyway, it was very useful to have found this. And the other thing that's kind of satisfying about it is that the chemistry to make this leaving group fits perfectly into a scheme for actually building up nucleotides from small starting materials. So you can make this and the key intermediate on the pathway to making nucleotides in the same reaction mixture. Right? So that makes it all seem like, bits and pieces of the puzzle are starting to fit together. And then more recently, there's been an even better synthesis of this material worked out in John Sutherland's lab in the UK. Okay, so we've been playing with these molecules for a while and trying to see how we can use them to do better copying chemistry. And so what I have to, this is a little bit complicated, but I have to explain it because it turns out to be really important later on when we start looking at alternatives. So for at least 50 years, everybody in the field, including me, assumed that the way to copy a template in a prebiotic fashion without enzymes was going to be basically the same as what happens in biology. You have what we call a primer that's annealed to the template by Watson-Crick base pairing, and then activated nucleotides come in, they bind to the template, sit next to the primer. The 3' hydroxyl at the end of the primer attacks the adjacent phosphate, and the primer grows by one nucleotide, right? Which is what biology does, except using enzymes and NTPs. And so it turns out that's not actually how it works, which was a total shock to everyone. What actually happens is that nucleotides like this, they're pretty reactive. They can react with each other and make this a really bizarre looking compound here, which for short, we can call it a bridged dinucleotide. Or if you hear me talking about bridged species, this is what I mean. So the whole reason that this works better is because you make more of this, and this is more stable. And this type of compound can bind to a template by 2 Watson-Crick base pairs. So it binds much more strongly. And this bridge structure is perfectly organized in 3-dimensional geometry to favor this reaction. So it's like perfectly in line, and it makes everything work better. This turned out to explain a 25-year-old observation from Leslie Orgel's lab, which was so puzzling, people didn't even want to think about it for decades, right? Which is that if you have a primer and you set up this experiment so that only one nucleotide can come in, it basically doesn't work. But if 2 nucleotides can come in, all of a sudden it works. So there was a catalytic effect that everybody just ignored because they didn't know how to explain it. It wasn't until Travis Walton joined my lab that he figured out that this is the chemistry that explains, I'm not sure if you can see this, that explains this whole effect. You have to make this intermediate, covalent intermediate, in order to catalyze template copying chemistry. Okay. All right, so let's see. So we've worked on that in quite a lot over the last 5 years. So it's a lot of detailed reaction kinetics, which I don't have to go into. But I couldn't resist showing this slide because this is one of the most satisfying things I've ever seen in my whole career, which is a series of X-ray crystal structures. Basically, what was done, all by Wen Zhang, a former postdoc, was to take 2 pieces of RNA that will base pair with each other in the middle and have a little overhang at the end that is a template. Okay, and we can actually grow, or not me, but Wen and other people in the lab can grow crystals of those RNA molecules with an unreactive nucleotide bound to that template region. Okay, so we harvest lots of those crystals, then we take those crystals and soak them in a solution that contains the activated monomers, for example, in this experiment. And after just 15 minutes of soaking, the original nucleotides that were bound to the template, which is up here, are gone. And our 2-aminoimidazole nucleotides have replaced them, and we see them bound to the template next to the primer. The purple stuff is the primer. But the actual leaving group is totally disordered. It's floating around in the breeze. We don't see it in the crystal structure. Okay? And initially, in our early experiments, that was really frustrating because we were thinking there should be some non-covalent interaction that stabilizes everything in the right conformation, and it just didn't work. But in this particular type of experiment with these activated monomers bound, if you just wait, and after about an hour, you can see there's new electron density between the 2 phosphates, right? So this is that bridged structure that I showed the chemical structure of earlier. So it's formed in the crystal. And if you wait longer and put in a catalytic metal ion, what you see is that this electron density in the middle goes away, and you see new electron density in between the end of the primer and the adjacent nucleotide. So this is the actual primer extension step, the formation of a new phosphodiester bond. So it's the elementary step in copying a long template. So we can see the whole process happening in a series of crystal structures. So that was a very satisfying advance. Okay, let's see. So there's a few other things that I want to tell you before we get into the question of why is this all happening with RNA, and could it happen with other things? So one thing is what I've showed you so far is 2 monomers bridged to each other. A monomer can also be bridged to longer bits of RNA, say 2, 3, or 4. And if you do that, surprisingly, the reaction rate goes up if the downstream part is longer. And not surprisingly, the affinity for the template also gets better and better. So these things are starting to teach us how to more effectively copy short bits of RNA without enzymes. Okay. And let's see, this is just an example of what we can do these days. This is actually from Lee's original paper. But this is a time course of copying a template without an enzyme. Okay, so you start off here at 0 time. This is a labeled primer. Things have been run down on a gel. After 30 minutes, you can see longer products appearing. Reaction continues. After 18 hours, we're up to +7, right? So not very impressive compared to what happens in your cells like all the time. This could happen in milliseconds if you have enzymes, but this is just chemistry. So we think this is kind of the underlying chemistry for how to copy RNA. Why does it stop here? It's because all these things are very reactive. They're sitting in water, they're hydrolyzing. The whole system kind of gradually grinds to a halt. One of the big puzzles in the field is how do you bring chemical energy into the system and keep it going? So there are some recent ideas on that, a lot from the Southern lab again. And so that's something else we're working on. But the idea is, if you keep bringing chemical energy into the system, you can kind of keep this process going and copy longer and longer sequences. Okay. All right. One final thing is we're not entirely in the dark ages of running gels and stuff like that. Daniel Dostovich in the lab has worked out a way to use next generation Illumina sequencing so that we can have a random template region, come in with activated monomers or bridge species, do some copying, and then sequence everything. That way we get to see on every possible template, every possible product, all possible errors, the context in which the errors happen. And it's giving us a lot of insight into how to do the copying more accurately and to get more efficient copying overall. Okay, so with that as background into copying chemistry, a very important point that was recognized again by Leslie Orgel and also Jerry Joyce a long time ago is that all these kinds of experiments I just showed you are completely ridiculous from a prebiotic chemical perspective, right? There is not going to be, a nice prebiotic pool on the early Earth filled with high concentrations of absolutely pure activated ribonucleotides, okay? It's very nice, and you have to start off in lab experiments with this kind of artificial pure systems, but come on, we have to think about how to make this more realistic. How could something like this actually get started on the early Earth? Okay. So if you start to think about what else might have been around on the early Earth, and looking at all the chemical synthesis pathways, for example, and looking at some of the properties of RNAs, it starts to look more complicated and messy. And so this is just the tip of the iceberg as to how messy things could be. If you look at the nucleobases, right? In modern biology, we have A, G, C, and U. Of course, there are hundreds of modifications that are found in mRNAs, tRNAs. But were those, there are all kinds of variant nucleotides that would have been or might have been around. And what would that do to this effort to get copying off the ground and in a way that you can inherit information? If you look at the backbone of modern RNA, enzymes make linkages between the 3' hydroxyl and the 5' OH on put a phosphate on the 5' hydroxyl. That's 3', 5' linkages. But if you do it without enzymes, turns out that 2' hydroxyl is more reactive. So you often make this wrong linkage. Okay? And on top of that, there's all kinds of other sugars, right? RNA uses ribose, hence the name. But there's like a gazillion other sugars that could arise depending on how you make them. So why would you start with just the ingredients for modern RNA? And actually, it's even worse than that. So this whole question of the early heterogeneity of the chemistry led a lot of people to think that starting biology with RNA was just chemically unrealistic and impossible. And so maybe life started with something much simpler or easier to make or more stable. And then that led to an explosion of really interesting chemistry. And I think the high point of that was work done in Albert Eschenmoser's group, I think one of the great chemists of our time. Albert actually chemically synthesized all kinds of variations of different sugars, different nucleobases, and started to look at their properties. And it turns out, RNA and DNA, that make a nice Watson-Crick base pair double helix with anti-parallel strands, that's not something unique. There are lots and lots of variations that do the same thing just as well. Sometimes with stronger base pairing, sometimes with weaker base pairing, sometimes with different base pairing rules. All the backbones structures drawn with red can base pair with each other. This yellow one is an outlier. It only base pairs with itself, not anything else. The blue guys here in this column can all base pair and talk to each other, but not with any of the others. There's this huge possibility out there in chemical space. All these things look like they could be perfectly reasonable genetic materials. So what's special about RNA? Was it just some historical accident of the particular properties at a particular time and place that led to biology starting with RNA? Could it equally well have started with any of these alternatives? That's been an open question. That's been something I think we've kind of accidentally contributed to having at least an idea about. Okay, so there are too many possibilities out there to study everything in detail. And so what our approach has been is to look at the kinds of molecules. If you were trying to make RNA, what else would you make by accident? Right? So you have to look at things that are close in structure. And that's what we've focused on so far. Okay, so here's the structure of RNA. Here is a very close relative called arabinonucleic acid, or ANA. The only difference is the hydroxyl here that points down, points up in arabinonucleotides. So it's pretty similar. And over here is something called threose nucleic acid, or TNA, where the sugar has 4 carbons instead of 5. Okay. And so I want to go through in some detail why we're looking at these as possible alternatives and why we think they wouldn't work. All right. Okay, so here is just another look at those alternatives, focusing on the activated building blocks, the nucleotides. Okay, so again, they're very similar in overall structure. It's just either this hydroxyl points down or up, or this carbon is missing, and you have a smaller sugar here. Okay. All right. So yeah. So why do we think these are interesting, and what's the real issue? So if you want to build a sugar that has 5 carbons, like ribose or deoxyribose, the best way to do it is to take a 2-carbon unit and a 3-carbon unit and put them together. 2 + 3 = 5, right? Simple. It's a little, well, so there are two important things. So chemically, you can do this. It works pretty well. You make these intermediate structures, but you get these 2 stereoisomers in roughly equal amounts. This is the one that would go on to make the arabinonucleic acid. This is the one that would go on to make RNA. They both get made. So, okay, what's to distinguish them? It turns out that this intermediate crystallizes very beautifully. And actually, I'm not sure if that's gonna, yeah, you get these incredible crystals of it. But the question is, do you actually want to use crystallization as a way of purifying an intermediate? That actually is a pretty nice idea. Or do you want to crystallize out a byproduct and go with what's left in solution, in which case you could make ANA? Okay, even if you crystallize out this and you're going to go on and use this to make RNA, some of this stuff is for sure going to be around, and you can actually interconvert these products. So I think if you're going to make RNA, it's almost unavoidable that there would be some of the arabinonucleotides around. Okay. All right, but the trick with this pathway is you have to come in first with the 2-carbon sugar, okay, do its reaction, and then come in with the 3-carbon sugar, glyceraldehyde. And that's a little tricky, right? How's that going to happen on the early Earth? So it turns out there are some possibilities, but wouldn't it be simpler to just go twice with the 2-carbon sugar, not worry about having to do sequential additions of different starting materials? And so if you do that, you end up making, 2 + 2 is 4. You make a 4-carbon sugar, and that can go on and make this threose building block and threose nucleic acid. Right? So that actually looks easier to make. And the discovery of TNA was made in Albert Eschenmoser's lab. And it really created a huge uproar because, yeah, it looks easier to make, and it actually makes a Watson-Crick base pair double helix, and it can base pair with RNA, it can base pair with DNA. It looks like man, maybe biology started with TNA and then did a transition to RNA and DNA. Okay, so we have to look into whether that was possible or not. All right, so here's the situation. We imagine our warm little prebiotic pool on the early Earth, and it's filled with nucleotides. And don't worry too much about the details there because we haven't still figured it all out. But at least there's going to be, if there's ribonucleotides, there's also going to be arabinonucleotides, and threonucleotides, and probably deoxynucleotides, and probably all kinds of other stuff, okay, which we're not worrying about for the time being. If all these different nucleotides, they can start reacting with each other and spontaneously building up short oligonucleotides, short chains that are random in sequence and also random in composition, okay? And that does not look like a very good starting point for Darwinian evolution, right? It's so heterogeneous, and if you can't reproducibly make a structure, then how can you ever get anywhere interesting? Okay, so that was the problem. All right, so where we got into it is we'd been studying copying chemistry for a long time. So we started to say, well, maybe they have some differences in how well they do copying chemistry. Also, not a new idea from us. I mean, Leslie Orgel and many other people have been looking at this much earlier on. But back then, the copying chemistry actually wasn't properly understood. So we thought it'd be a good idea to revisit some of these early experiments. And so what we could do is, now that we understand like how RNA copying actually works, is we can look at every step of the process independently and compare it with the building. How well does it work with the building blocks of RNA? How well does it work with the building blocks of ANA or TNA? What happens if you mix everything all together? Does the system fall apart? Does it get poisoned? What happens? Okay, so we started off looking at ANA. And so a lot of this, this work was done by Chris Kim when he was a graduate student in the lab. And so the steps that he looked at were, okay, if you make ANA nucleotides, you activate them, you mix them in solution, do they make this bridged dimer? And they do. That step's not really effective, okay? If the bridged dimer binds to an RNA template, and you have an RNA primer here, how does the primer extension step happen? And it's slower. It can be actually quite a lot slower. Okay. It turns out if you mix ribonucleotides and these arabinonucleotides and do a competition experiment, what you make is almost entirely RNA extension. But okay, there's still some reaction. So what would happen if an arabinonucleotide gets added to a primer? It turns out that's really slower. That's almost like a chain terminator. Okay, what happens if the arabinonucleotide's in the template strand? Actually, that's not such a big deal. You can copy over that variant nucleotide and make RNA. All right, so we make all these observations, we measure binding, we measure rates, we want to understand, like, why is it different? And so one of our, so this is how we do the kinetics, by running gels and measuring rates. Yeah, we can put the variant nucleotides in different positions. And so, yeah, for example, here, if the variant nucleotide, the arabinonucleotide's at the end of the primer, extending it, nothing happens. If if this is a ribonucleotide, you see primer extension. If it's an arabinonucleotide, it's almost dead. So those are the kinds of experiments that we typically do. But then why is that happening? And so to get at that, again, we go back to X-ray crystallography. So we grow, we chemically synthesize these chains that have the variant nucleotides in different positions wherever we want, grow crystals. Sometimes it works, sometimes it doesn't. It turns out if you put an arabinonucleotide in the position that would represent the end of the primer, and grow crystals with the bridged RNA species next to it, you can see that this distance from the 3' hydroxyl to the phosphate that it should attack is about 2 angstroms further than in the case where this is all RNA, and like in the series of crystal structures that I showed you before. So it turns out that when you make that arabino nucleotide, the sugar is in a different conformation, and that different shape pulls the 3' hydroxyl away from the phosphate. It's farther away, so the reaction is slower. Okay, it's a simple potential explanation. Okay. Yeah, like I said, if you put the arabino nucleotide in the template, it's not too bad. You can still copy it. It all works pretty well. Okay, so the basic idea is that you can copy over these things. You make RNA. If you incorporate them, it's bad. And actually, the incorporation is slow. What about TNA? This is even more interesting because this is the one that looks like it might be easier to make, right, prebiotically. So again, we go through the same kind of sets of experiments. We can make activated 3-O's nucleotides. We measure the rate at which they form the bridged intermediate, and it's slower this time, okay? Like 4 or 5 times slower. Then when it is bound to the template, what about the primary extension reaction? That's also slower. Okay. What happens if one gets incorporated and you try to extend beyond that? That's also slower. So every step of primary extension is slower if you use these nucleotides that we thought might be easier to make. On the other hand, again, if that variant nucleotide is in the template region, yeah, you can copy over it and make RNA. And again, so the idea is that these things would be filtered out at various steps along the pathway. Okay. Let's see. Again, we want to understand why TNA is worse than RNA. So again, go back to growing crystals, trying to look at the structures. And so the thing that makes TNA so much worse is the fact that it's missing this 1 carbon atom here. So the phosphate, instead of being held out away from the sugar, where it's sort of poised to react, it's held like in close, right over the sugar. So it's very crowded in there, and it's hard to make the reaction go. Okay? And also, because of the fact that it's held, the phosphate's held over the sugar, again, the hydroxyl at the end of the primer is really far away from the phosphate that it should attack. Right? And so the chemistry is much slower. Okay? All right, so here's, let's see, yeah, here's what happens if you put a 3-O's nucleic acid, nucleotide in the template. Again, you can copy over it, very little effect. Okay, so so why is that? Again, we can look at crystal structures. It turns out that the sugar phosphate backbone of RNA is so flexible, right? There are so many rotatable bonds that you can, or RNA can accommodate this kind of structural perturbation with only local changes. And the changes are very, very minor. Right? So it totally makes sense that you can copy over this kind of heterogeneity in the template. Okay. So what about other kinds of heterogeneity in the template? I mentioned at the beginning a few other things that can happen. So RNA is all 3', 5' linked when it's made by, say, RNA polymerase. But okay, you're doing this chemically. What happens if you make a mistake and there's a 2', 5' linkage? Well, it turns out you can copy over that linkage. And then using enzymes, we can see, is the product 3', 5' linked or 2', 5' linked? Turns out it's almost always 3', 5' linked. So you have a heterogeneous template, you copy it, and you get what looks like modern RNA. Kind of magically comes out of the structure. Again, we can grow crystals and see kind of why that is. The structural perturbation of a 2' 5' linkage that's shown in here is so minor that, unless you're really used to looking at these structures, you can't even tell. Right? It's so close in structure. All right. Another kind of heterogeneity that might be expected is, well, what if you have 2 phosphates instead of 1 phosphate in the backbone? And there are chemically reasonable ways why you might think that would be almost inevitable. Again, the backbone can accommodate that, and all the other changes are very minor. So you can copy over a pyrophosphate linkage, again, make normal RNA. Works fine. Okay, so you put all these experiments together, you get a model like this. This is how we're thinking about it at the moment. We have our nice warm prebiotic pool full of activated nucleotides, very messy mixture of different kinds of nucleotides. They condense to make random sequence short oligos that incorporate this heterogeneity. And I should say, this is also an important step, right? What gets filtered out at this step? So there's a graduate student, Xuan Jia, working on this in my lab now. And it turns out the 3' nucleotides get a little bit filtered out at this stage. The arabinonucleotides, not at all. Deoxy we haven't looked at yet, but there's some interesting things happening there. Okay, so you make these heterogeneous oligonucleotides. They're random in sequence, right? So the idea is every now and then, there'll be little bits of complementarity. So these oligos can find each other and base pair. Now you're set up to do primer extension chemistry. And when you do that, it doesn't matter what's in the template, you're going to make RNA. So a few cycles of that, you keep iterating that process, and you'll end up making oligonucleotides that look more or less like modern RNA. That's the theory anyway. That's our hypothesis. And so we think that provides a way that you can get to a relatively homogeneous set of starting materials that would allow reasonably accurate replication to go on, and then you make sequences that could fold up into structures that could do useful things like catalyze reactions, so ribozymes. And that would be now reproducible, right? Because the molecules you've made have a homogeneous backbone and homogeneous chemistry. And so that would kind of help a lot to get Darwinian evolution off the ground. Okay. Right, so basically we think that, yeah, life started with RNA, and basically it's because RNA always wins. If there's competition, RNA wins. There are other steps on the pathway that also contribute to this. So a lot of variant nucleotides get filtered out by UV irradiation. So the early Earth, no oxygen, no ozone layer, intense ultraviolet. It's actually needed to drive synthetic chemistry, but it basically helps to get rid of a lot of stuff that you don't want that to be labeled, destroyed by UV. Of course, the synthesis also matters. It turns out that so far at least, the chemistry going to ribonucleotides looks kind of favored, predisposed. This part of it, it's hard to know if it's because what people have studied so far is how to get to RNA, and how to get to a lot of other things has not been looked at as much. But there are aspects of it that make you, if you put all this together, is it really true that there's only one way to get life started, and that's by going to RNA? That makes me very uneasy, right? Why? It seems more likely that there should be 0 ways or lots of ways for something to happen. So it's kind of, I don't know, it's an interesting philosophical point. If we find life elsewhere, some exoplanet someday, is it also going to be the same basic chemistry because that's what chemistry leads to everywhere? I don't know. Interesting question. Okay, and our little contribution has been that the copying chemistry works best with RNA. Okay, so all this was done by fantastic grad students and postdocs in the lab over the last many years. I've tried to mention some of the key people as I went along. Lee and Travis figured out the basic copying chemistry. And Chris did a lot of the work looking at alternatives to RNA and trying to explain why they get filtered out. And so I'll stop there. Happy to answer any questions.
Okay, I'm going to moderate a question and answer session, and then following that, we'll have closing remarks by Conrad Gilliam. We're on Zoom here, so there's also Amanda and Nick will monitor those questions. But when there is a question, Jack, I'd like to ask you to repeat the question so that the Zoom people can hear it.
Yeah.
And the committee has requested that the first couple of questions come from students. So Jack's happy to take questions from students initially.
Yep. Yeah.
First of all, thank you for giving. My question was, what is the hypothesis for where?
Right. So the question is, where did all these nucleotides come from, right? How are they made on the early Earth? And so this is a very long-standing question, a lot of very complicated chemistry. There are lots of different theories. My personal favorite is a very detailed set of pathways that have been worked out in the Sutherland lab in the UK. And the idea is that life starts with cyanide. So I love that because it's so counterintuitive. Cyanide is, there's a lot of energy stored in it's a very reactive compound. So you can basically start from cyanide and build up all of the nucleotides, sugars, even parts of lipids, and most of the proteinogenic amino acids. You can get everything out of cyanide. And there are some other beautiful aspects of the chemistry, but that's kind of, I think, the best developed insight into how it all got started.
Another question from a student?
Yeah.
RNA molecules, why could the primordial cell want that instead of keeping a full of short?
Right. No, that's a great question. So would there be some reason, some selective pressure, or I guess chemical issue that helps you get longer and longer oligos? So that's a great question. If you want to make a catalytic RNA, you need a certain length for it to fold up into a nice three-dimensional structure that'll do something interesting or useful. So for a long time, we were thinking about, how can we do that, right? How do we make oligos that are 50 or 100 long? And copying things that long, and especially things that fold up, turns out to be really hard. And so that's what's driven us to think more about, can you do replication with short pieces and then somehow assemble the short pieces into longer things that can be catalysts, for example. So that's kind of how we're thinking about it. And then of course, later on, once you have, say, ribozymes and more effective replication machinery, then you can make and copy things that are longer, which has lots of advantages. But I think that would be a hard way to get started. Yeah. Right. Okay. Lots of questions there. Okay, the basic question is, well, yeah, what about the membrane part of a protocell? Why do you need it? How would it form? What do we know about its properties? And so the basic idea, the underlying idea, is you need compartmentalization in order to have Darwinian evolution. If everything's floating around in solution, you know, a molecule that does some really fantastic reaction can't really benefit itself. Right? It might say you make some fantastic product, diffuses away, helps everybody else. You need to close that feedback loop to have Darwinian evolution. The way to do that is to have all this going on inside compartments. But then, of course, the compartments have to grow and divide. And so there are alternatives, right? So for continuity with biology, I think membranes is the way to go. And it turns out that some membranes, it's really easy to make them grow and divide. There's a lot of interest in phase-separated, liquid droplets as an alternative way of doing compartmentalization. You can't rule it out, but to the extent that we've looked at it, RNA molecules tend to exchange fairly rapidly between different phase-separated droplets. And so over time, they're not really individual compartments. So that's why we've been exploring membranes for a long time. Question is, okay, what are you going to make them out of? Has to be something simple, has to be available. So on the simple direction, fatty acids are a great model. The problem is nobody really knows how they could have been made on the early Earth. There's some different theories. None of them are very convincing. So it's one of the big open areas in prebiotic chemistry that needs a lot more work. Yeah, I hope I answered at least some of your questions. Okay. Yeah, Shannon. So, we're just trying to let the chemistry talk to us, right? If we had found, for example, that TNA is just as good as RNA or better, then I would be telling you about how life got started with TNA and transitioned to RNA. But it turns out, we've really tried to look at that and say, okay, like the experiments I showed you were a system that's mostly RNA and we put TNA in different places. That doesn't work. Okay. What if you made everything out of TNA? Turns out that doesn't work at all. That is totally dead. So I think chemistry, we just have to let the chemistry tell us. So far, I can't rule out the possibility that there's something we haven't looked at that might be the magic alternative, but so far it just looks like the easy and most effective way to get going is through RNA.
Yeah, we have a
Yeah.
So maybe could there be considerations that there's a different leaving group
Yeah.
that would then allow for the DNA
Yeah.
to have better
Yeah, no, that's a great question. Did you review our paper? Yeah, as something that I can't really rule out. I don't know anything in talking to other people. I haven't come up with, I haven't heard a good alternative that might make it work better. I think the limitations of ANA are more in the intrinsic geometry of the sugar. So ANA, for example, can't even base pair with itself. It can base pair with RNA and DNA, but not with itself. But I think the problems with ANA and with TNA are intrinsic to the structure. And so there is a kind of way to do what you're suggesting. If you make an aminonucleotide version of TNA, so now the nucleophile is much better, leaving group's still the same. You can sort of crank up the reactivity and make things work a bit. With enzymes, we can also make it work a bit. So you actually can do lab evolution with TNA using enzymes. And maybe this amino TNA might be an alternative, but the prebiotic synthesis of that doesn't look great to me. So yeah, we just have to keep exploring different variations, see what works, what doesn't. But yeah, great question.
So we have,
Jack, we have a question from the Zoom.
Yes.
Paul mentions that you describe RNA as stable, but it's often described as unstable, at least relative to DNA. Is that stability only in the absence of enzymes? So can you talk about sort of what stability means and maybe how that's relative to the stability that we're familiar with in today's biology?
Yeah, okay. So you know, RNA is notorious for falling apart while you're trying to work with it. And most of that is because, you know, you have to be really clean, and there are ribonucleases everywhere that degrade RNA. But, you know, if you get rid of that and you are clean, you can, you know, it's not that bad. But if you have transition metals around, like iron, for example, that's really bad. That'll chew RNA up totally, very quickly. And so then you have to say, okay, in our like warm little prebiotic pool, how are we going to protect RNA from degradation by iron? And it turns out cyanide to the rescue, right? Free iron is complexed very tightly by cyanide, and that would protect the RNA from degradation. Now, you can argue that being really, really stable like DNA might allow it to accumulate even if it's made less efficiently, right? And it might predominate even though its copying chemistry is worse than RNA. So yeah, that's a little bit hard to argue against, but you know, is the ability to degrade RNA an advantage or a disadvantage? And in terms of being able to recycle stuff, it could be a huge advantage, right? Once you make something in DNA, and as say if you make a mistake while you're making something in DNA, it's like set in stone. You're stuck. Whereas with RNA, you can degrade it and start over again, in principle. Yeah. So that's the long answer. The short answer is we don't know. We have no idea.
any other Zoom questions?
Is also wondering about the concentration of monomers in reactions and how it would compare to the primordial soup, even if you had.
Yeah, so okay, question of the concentration of the building blocks. And so this is a great example of in the early experiments, like when Leslie Orgel started working on this stuff in the early 70s, late 60s. You would have to use really high concentrations, like 100 millimolar, 200 millimolar, to drive any assembly and copying chemistry. And that always seemed like a stretch, to put it mildly. Okay. And so stepwise, I think we're in a more realistic regime now. So having these bridged species lets everything work with, you know, worst case low millimolar, often high micromolar levels. If you use monomer bridge to oligos, you can make everything work at submillimolar concentration. So I think that might be more realistic.
So according to the schedule I have, we have time for one more question.
Yeah. In the affecting the copying chemistry? Or yeah, so okay, so question is a role of lipids or membranes in sort of helping some of this chemistry to go on. So yeah, we've played around with various aspects of that. One thing we were super excited about for a while is what if we could make everything happen on the surface of a membrane, right? And then in theory, you know, everything should work much better, right? Because more collisions, you know, the reduced dimensionality should be great. We're never able to make that work. So it doesn't, maybe we just didn't do it the right way, but it's an interesting possibility. at a little more advanced level, Irene Chen has done a really beautiful experiment looking at the evolution of ribozymes in solution versus in compartmentalization. And she found that let me explain this, the variation in activity is greater when you're encapsulated than in solution. And that means because you have more variation, more phenotypic variation, that selection can have a stronger effect. And she can actually see an acceleration of Darwinian evolution with compartmentalized RNAs compared to in solution. So yeah, so we love that, and we're hoping to work with her on that when we get to that stage.
And we'll do a final question. Yes, go ahead.
Yeah. Oh, PNA, peptide nucleic acids. Yeah. So that's another variant on informational polymers that was really exciting for a long time. Still exciting for technological applications, but I don't think I have ever seen a prebiotically realistic scenario for generating PNA and getting this to start. So yeah, I don't see any way to get going with PNA, really. Yeah.
Okay, well, let's thank Jack once again.
Thank you.